Hello, my name is Rudy Summers, and my microbiology assignment is on Streptococcus pneumonia. So, first off, starting off, like I said, it's a pneumococcal disease, and has a scientific classification as a domain as a bacteria. Its class is the bacilli. The order is Lactobacillus. Family is from the Streptococcaceae. Genus Streptococcus. And the species is also Streptococcus pneumoniae. Transmission for this disease is person-to-person transmission of the Streptococcus pneumoniae. Occurs via droplet transmission through expelled respiratory secretions and is relatively common. The bacteria are considered normal flora of the human nasopharynx, and only rarely do the bacteria cause an infectious disease. Nearly all children harbor one or more of these strains and become carriers during their first year of life. The human nasopharynx is the only naturally existing reservoir for this Streptococcus pneumonia. The Streptococcus pneumonia is also a classic example of a highly invasive gram-positive extracellular bacterial pathogen, major cause of morbidity and mortality globally, causing more than deaths than any other infectious disease. Some of the characteristics that we'll have, it's you can only view it microscopically. Cells are lancet shaped, ranging in diameters between 0.5 to 1.25 micrometers. Most often they're seen in a diplococcal arrangement, but can also be found singly or in short chains. It is gram-positive, biosoluble, and aerotolerant. They are alpha hemolytic, but under anaerobic conditions become beta hemolytic. This is caused by an oxygen-labeled hemolysin. Changing colony morphologies arise from the Streptococcus pneumonia unique ability to self-destruct. Nearly all clinical isolates make the morphologies arise from the pneumonia's unique ability to self-destruct. Nearly all clinical isolates make the enzyme autolysin and undergo the lysis within 18 to 24 hours of growth and initiation. If the growth is stationary phase in the lab, the entire culture will be killed. Initially, the colonies are seen with a plateaued elevation, but this later collapses in the center, signaling the autolytic event. It has been suggested in one study that the fragments of autolyzed bacteria protected the other still living bacterial cells from phagocytosis. A reduction in the phagocyte-mediated elimination of the pneumococci from human blood was observed. These infections may also be diagnosed from symptoms, x-rays, tissue specimens. Laboratory identifications include the gram staining of sputums, blood, cerebrospinal cultures. It's also differentiated from other streptococci by quelling reaction, biosolubility, and apoptosis inhibition. Some of the signs and symptoms. Infected persons experience a sudden onset of illness with fever and malaise. These symptoms are common to all forms of pneumococcal infection. And in the pneumonia, symptoms include cough, chest pain, and sputum that is purulent or blood-tinged. Symptoms may present less abruptly in elderly. Sputum may be absent, and additional symptoms may include shortness of breath or altered mental status. In pneumococcal meningitis, symptoms are a stiff neck, headache, lethargy, and seizures. In otitis media and sinusitis, Sinus and ear pain are typical symptoms. Some of the virulence mechanisms is considered to be the primary virulence mechanism of the pneumonia bacterial capsule. It interferes with the phagocytosis by interfering with within the binding of the complement of C3b and its surface. Capsulated strains are 100,000 times virulent than those without capsules. The pili present on some isolates are responsible for initial adhesion, providing that the subsequent ability to become invasive. The peptoglycan and the teichoic acids complex of the cell wall is highly inflammatory. Serous antigens, phosphoglycines, and lipoteichoic acids are factors that enable adhesion and invasion. Additionally, the pneumonia produces enzymes such as neuraminidase and IgA protease, which increase in virulence. And during the meningitis, the bacteria's production of hydrogen peroxide can cause apoptosis in neural cells. And treatment of the infection and pneumonia are usually treated with penicillin, but other antibiotics may be substituted in case of drug resistance. And pneumonia is one of the main diseases that's caused by the Streptococcus pneumonia. And different forms that we do have, I mean, that the Streptococcus does cause. I mean, you got strep throat, scarlet fever, erysipelas, puerperal fever, and some of the pneumonias. And again, like some of the drug choices that are used for healing or treating this infection is penicillin, and another choice would be erythromycins. Prevention of bacterial pneumonia is vaccination against the Streptococcus pneumonia, the pneumococcal polysaccharides vaccine for adults, and pneumococcal conjugate vaccine for children. And these are the two types of injections or vaccines that we do have. And vaccinations, persons for whom pneumococcal polysaccharide vaccine is recommended, all persons aged over 65 years of age, persons aged 2 to 64 years with chronic cardiovascular pulmonary disease, functional or anatomic asplenia, cerebrospinal fluid leaks, immunocompromising illnesses such as HIV or AIDS, malignancies, chronic renal failure, necrotic syndromes. Organ and bone marrow transplantations also are included in that.